Okay everyone, welcome to my YouTube channel. እንኳን ደህና መጣችሁ። ዛሬ የምናየው ክሊኒካል ሙቭ ኤግዛሞች ላይ ያሉ ኤግዛሞችን ነው። what we have to do, how you can manage our time, so what kind of questions are usually presented on move exams, we will see in detail. So try to watch until the end. Don't forget to subscribe and share it to your friends and like the video. So we'll begin. So try to watch with VPN as much as possible. በቪፒኤን ለማየት ሞክሩ። ያው ቻናሉን ማበረታታ ስለሚሆን እናንተ መረጃ እያገኛችሁ ቻናሉን ታበረታቱበታላችሁ። ሶ በቪፒኤን እንድታዩ እንመክራችኋለን። So let us see the general directions. Okay, the general directions. So move exams are usually practical exams, and sometimes some of them will present the theoretical parts, but usually we will be asked the practical ones. Okay, the practical ones we may be requested to demonstrate. Okay, we may need to be requested to demonstrate the actual procedures, especially diagnostic procedures, microscopic exam reports. So for the general requirement, for the general direction, as a general direction, usually for each exam, for each equations in every section, we are given 2 minutes in every exam, usually 2 minutes. So we have to manage our time. The second thing is from each section we will be asked usually the practical part. We will be asked the practical part. The third one is usually 2 up to 4 questions will be presented for us when we are on move exam. We will be given 2 up to 4 usually, minimum 2 up to 4 from each section. Okay, usually for sections such as hematology, clinical chemistry, bacteriology, usually we will come across from 2 up to 4 questions, but for others we may face 3 or 2 questions. So usually the questions in each section are 2 up to 4. The other thing is we have to stay calm. Okay, the major thing is to avoid anxiety. Okay, we have to avoid anxiety during move exams, because if you don't avoid anxiety, it may be difficult for us to recall. Okay, to remember things we studied. Okay, remember things we have done in the laboratory session, on our back room or clinical practicum session. So the major thing is managing stress. We need to manage stress. The key to success on move exam usually is, in every exam, is managing our stress, managing emotional tension. Okay, so let us go to the exam. So you should know that this may not be the exact questions in every section. We'll see the most common questions, that usually be presented, but this may be the most common. Okay, so the first section will be bacteriology. So usually bacteriology, the first thing, the most common thing is we will be given culture medias. Okay, we'll be given culture medias, and we may be asked to identify them. Okay, to identify them, which kind of culture media, and also their purpose. Okay, so we may see some culture media. We may be given culture media such as mark, nucleate. So we have to then name, and we have to name the culture media, and we have to identify its purpose. We have to state our purpose. Mark, okay, what kind of media is it? What is the purpose of mark? Blood agar plate, Muller intan agar. So we may be given the culture media. This is very common. Okay, you have to know this. So we have to know at least the major culture media, their contents, what kind of contents are in mark, in BAP, or in chocolate agar, or other medias, and we have to identify the purpose, their content. This is very common. Okay, so the second thing is we have to identify the difference between fluorescent and light microscope. This is also common, very common. We have to identify in terms of sensitivity, specificity, as well as the resolution and the magnification behaviors. For example, fluorescent microscope how uses what kind of light? UV light, right? But light microscope usually uses the ordinary light. And fluorescent is usually used with fluorescing stains such as oramine, right? For mycobacterium diagnosis, we use oramine for staining. And in light microscope, we use ordinary stains, right? Ordinary stains such as crystal violet, carbon fusion, and others. So we have to identify in terms of their magnification capacity, their staining behavior, for what kind of microorganisms do we use the light microscope and the fluorescence. This is also very important thing. We have to identify which microorganisms are identified with light microscope and with the fluorescent microscope. Okay, so this is very important. Another thing is we have to identify and report the bacteria on a slide. This is also very common. Okay, we may be given some slide and we may be asked to report. Okay, we may be asked to report what kind of bacteria we'll see. So we have to follow the correct reporting format for every microbiological identification. We have to follow the correct report format. For example, when we see some bacteria, we have to what? We have to state their number, right? First, few, many, right? And their gram reaction. Next will be the gram reaction. Then what? Their genus and shape. Staphylococcus, streptococcus, and scene. So we have to follow this format. Many gram positive staphylococcus, few gram positive streptococcus. Okay, so we have to start from their number, then gram reaction, then what? Their genus, their bacteria genus, species. So we have to state just like we have to follow the correct format when you report. Okay, reporting is not enough, but we have to follow the format. This format is very important, and this is very common question or microbiology section of movie exams or clinical practicum exams. The other thing is the chemical used in staining. We may be asked some chemicals and their purpose. For example, what is the mortar used in AFB, acid fast bacillus staining? Okay, crystal violet. Why we use crystal violet? What is the purpose of crystal violet? What is carbon fusion in acid fast staining? What is crystal violet? Why we use crystal violet in the gram staining? Why we use iodine mordant? We have to identify the purpose of each chemical that we use in staining. Okay, not just mentioning, but we have to state the clinical significance or the clinical purpose of that stain. Okay, so this is also very important. Staining, microscopy, and culture medias, and sometimes we will be given biochemical tests, some biochemical tests, and we may be asked to identify their purpose. Okay, some biochemical tests will be served for us, then we will identify them. Okay, so this is very common, and also we may be also asked to measure the susceptibility and resistance behavior of some strains. We may be given some cultured media, and we may be given some drug or some disc, and we may be asked to measure the resistance. We may be asked to measure the zone of inhibition. Okay, the zone of inhibition, and we may be asked to identify whether that particular bacteria or particular microorganisms that have grown on that media is resistant or susceptible. Okay, so we have to identify. So this is important: the culture, the biochemical test identification, the purpose, and the antimicrobial susceptibility test. Okay, antimicrobial susceptibility test we may be asked to measure, and also the staining behavior and the report format is important on microbiology. This is very important. So don't forget to see this back. Okay, so these are important and most common in bacteriology. Okay, let us go to the next session. The next will be parasitology. So in parasitology, there are some things that are very common. The first thing is microscopic examination is very common. Usually very common. We always see these kind of questions we may be asked to identify the cyst of Giardia, amoeba, coccidium parasites. Okay, so we have to identify, especially for first year. We may be asked for identify the coccidium, the oocyst of the coccidium parasites, but usually on the first year or on the second year first semester, usually we may not come across this parasite, the coccidium parasite, but we have to also still identify them. We have to try to see them. Okay, so we have to identify them, and also the ova of elements from fixed samples, and we may be asked for stating diagnostic and the infective stage. Okay, right? For example, we may see some ova or the ova of Hooker, let us see. So we may be asked to state what we have seen on the microscope. Additionally, we may be asked to state the infective and the diagnostic stage of the ova. For example, for Hooker, okay, we see ova of cormicin. The diagnostic stage is what? The ova, right? And the infective stage was the filariform larva. So we have to identify the infective stage, the diagnostic stage of each parasites. Okay, this is very important in parasitology. Okay, the other thing is trophozoite or gametocyte of malaria parasite. This is also very common. Okay, we have to identify the trophozoite, the ring forms, the gametocyte, the schizont of malaria parasites, and we may be asked to state their species. We may be asked to state their species. This is also very common. Okay. Another thing is the manual calculation process, such as CATOCAS. CATOCAS is usually very common question in every movie exams usually. So we have to know the calculation. Okay. After counting, the number of ova we have seen on the 41.7 gram of the stool, we have to make a calculation. Okay, for calculation of the number of ova per ml. Okay, so we have to know the CATOCAS calculations. Not only CATOCAS. This is just for just stimulating you, for leading you the directions. Other calculations may be asked, but CATOCAS is very common. That's why I stated here. Another thing is concentration techniques. Formalin and ether, for example, we may be asked for the purpose of formalin in formoliter concentration. Formoliter. What is the purpose of formalin? What is the purpose of ether? Right? Ether is used to absorb the debris and to be on the supernatant. Okay, to absorb the debris and to suspend it on the supernatant. Whereas the formalin is what used to what is used to fix the specimen, fix the parasite or the ova, and to stay at the bottom of the solution. Okay, so we have to in every this is just for your example, in every concentration technique we have to know the purpose of each chemicals that are used. Okay, so this is important in parasitology section. Okay, another section is the sample reception and safety section. Okay, from the sample reception and safety section we may be asked different things. The first thing is symbol of biohazard. The yellow, okay, the red, the black, we have to identify which one is used for sharp infectious, for sharp non-infectious, okay, for non-sharp infectious, okay, and for non-sharp non-infectious. So we have to identify the type of biohazard and their purpose. Okay, and also the name. We have to maybe ask her to name at least 4 personal protective equipment or PPE. This is also very common. Another thing is what will happen or how long should the tourniquet be placed on the arm of the patient who is going to be drawn a blood, right? So we may be asked for how long it should be placed, and also what prolonged application will cause. We may be asked to state what does it cause when we place the tourniquet for long time on the arm of the patient, right? So hemoconcentration is a very common, right? So we may be asked this kind of questions. Okay, for your direction, we may be asked this kind of questions. Another thing is we may be given the paper, the needle, and the glove, and we may be asked to dispose them in biohazard garbage or in the yellow, black, and red garbage. Okay, so we have to know which disposal system is available, or is it uh, which is appropriate for which kind of biohazards, which kind of infectious things, or which kind of wastes we have to identify. Okay, so we may be given paper, glove, and for example, paper is what? Non-sharp, non-infectious. Glove is what? Non-sharp, infectious. And the needle is what? Sharp, infectious. So we have to know which biohazard disposal mechanism is appropriate for which kind of waste, laboratory waste. So this is very, very common. Another thing is we may be asked to define urgent specimen rejection criteria. Okay, this is also very common. Okay, we have to clearly state which kind of samples are urgent, why. For example, such as blood, CSF, they are urgent, right? Why? We have to name, we have to mention. Okay, we have to state. Other thing is, we may be asked to state the rejection criteria, sample rejection criteria. Okay, so this is also very common. Another thing is anticoagulant and their use is also very common question. The anticoagulant, we may be given the anticoagulant and we may be asked to state their purpose, which kind of samples, okay, which kind of tests are conducted from this anticoagulant. Okay, so we may be asked to state the kind of appropriate anticoagulants for what type of test. So we have to identify the purpose of anticoagulant for identified for laboratory diagnosis in different sections, for bacteriological, for clinical chemistry, for hematological. So we have to identify. This is also very, very common in the clinical practicum move exams for lab technologists or medical lab. Another section is hematology. So in hematology, the first thing is microscopic examination. Okay, so we may be asked to observe the specimen, the blood film or the blood smear, and we may be asked to state what we see. So when we are just asked to state what we see there, we need to report all of the things we see. We should not select any or everyone. Okay, we should not select one cell or one kind of special finding. We have to state all of the things that we see on that slide, on that smear, as much as possible. But if you are asked a special kind of, maybe special cell or abnormal cell, we may need to mention only that cell. But if you are asked to state all of the things that we see, we have to state all. Okay, with the WBC, platelets, abnormal cells. So we have to name all the things that we need. This is very common, and the very common mistake that usually the students make when attending the clinical practicum exams, because they are asked to state all of the things that they see on the slide, but they select to report only one. This is not right. We have to state all, even unless we are asked only to state only one cell. Okay, this is also very common. Another thing is hematocrit. We may be given a manual hematocrit material instrument, and we may be asked to measure. We may be given sample and we may be asked. So hematocrit is also very common, and we may be asked to state the principle of erythrocyte sedimentation rate. Okay, and it's anticoagulants used in Winthrop method, maybe. Okay, in Westergren method. So we have to know its principle, the type of anticoagulants used. Okay, so this is also very common. It's related with the anticoagulant use, very common. Okay, because some anticoagulants are used for some tests and some for others. For example, usually for WBC, CBC, we use EDTA. We have to know the background, right? We have to know the background. Why don't you use, for example, heparin for blood film analysis or for CBC? Why don't you use heparin? We have to know that. We have to identify type of anticoagulant used in hematology and why we use them. Okay, this is also very common. Another thing is we may be asked to prepare a blood film, a blood smear, and we may be asked to stain it. Okay, usually they will evaluate our blood film preparation, okay, and quality. So we may be given a mark based on our how much quality work we do with that regard. So blood film staining is also very common. Okay, we may be asked to prepare a blood film, and we may be evaluated based on the quality of the blood smear that we have prepared. So this is also very common. Another thing is we may be asked to state order of drawing sample. This is also very common, and also the storage of sample. So it's interdisciplinary actually. It's not only for hematology. This is usually asked in the reception usually, but sometimes in the we may find it in the hematology section because the order of draw. Okay, if you have to draw a blood with vacuum thinner from a certain patient sample, okay, we have to state which one should be drawn first, for which section, for which kind of test is drawn first, then next, and then after that. So we have to also now the storage temperature for every test. For example, we have to know the storage temperature for chemical chemistry, for hematology, for bacteriology. So the storage temperature of each sample should be identified. So this is also very common in on MOVE exams of practical, clinical practicum exams. Another thing is the hemocytometer counting and the cell concentration calculation. We may be asked to calculate the cell concentration. Okay, we may be asked to count manually the cells manually on microscope, and then we may be asked to do a calculation and report the results. So this is also very common. Okay, very common type of question. So we have to now the calculation of manual hemocytometer. Okay, so we count cells manually. We need to know the calculation step, the calculation. So this is very common. The next is lastly the clinical chemistry and urinalysis. So under clinical chemistry, the first thing common question is the pipetting techniques. Okay, we may be given a pipette, and they will or the evaluator will observe our handling and how effective are we in avoiding the air bubbles. Okay, so we have to know, and also reverse pipetting and forward pipetting. So we may be given some pipette, and the evaluator may ask us to do a forward pipetting and also a reverse pipetting. So we have to identify how the forward pipetting works. What is forward pipetting and how we can do forward pipetting, and how we can also do the reverse pipetting. So we have to know the steps, and also we have to know the proper handling of the micropipette, and also we have to train ourselves frequently to avoid air bubbles. We may be practically evaluated based on our pipetting technique on MOVE exams. Another thing is calculations. Okay, calculations, analytical calculations in clinical chemistry. For example, standard preparation. You already know that, right? C1 V1 is equal to C2 V2. So we may be asked to calculate the standard preparation or blank preparation based on this formula. So we have to identify, we have to know how to calculate the analytical calculation for preparing the blank solution, or and also for creating clearance, for example, UV over P. Okay, you know that the urine volume and the plasma volume and the urine volume of creatinine times what plasma flow rate. So this is also very common. So we have to identify, we have to know the calculation steps in analytical in the chemical analysis of the sample in clinical chemistry. Another thing is we have to know the normal reference values and their clinical meaning or their interpretation. Okay, for example, if bilirubin is high, what we expect jaundice, right? So we may be given some case. Okay, we may be given some concentration of bilirubin, and we may be asked to state the meaning, or we may be asked to interpret that concentration. Okay, so we have to know the background or the clinical meaning of each uh, concentration of analytes, and also we have to know also the reference range for every analytes, for bilirubin, maybe for enzymes, for lipids, proteins, for every we have to know the reference range. This is also very common. So, and also we need to know the clinical meaning of the concentration of the analytes. Okay, there is a very common question on practical movie exams. The next, the last one is urinalysis. So, or urinalysis section is usually given under clinical chemistry, but sometimes it may be separate, maybe given by its own. So, on urinalysis, we may be asked to do the physical examination for a urine sample. Okay, we may be asked to do the physical, the color, appearance, microscopic identification, and interpretation, also specific gravity. Okay, we may be given a urinometer. One time I encountered this question. I was given only urinometer, and I was asked to report the specific gravity. So you may be asked, maybe refractometer, depending on what kind of lab you are in. You may be given a refractometer or urinometer, and you may be asked to measure the specific gravity of your urine sample, and you will be evaluated. Okay, you may be evaluated. They will give you noun sample, noun the urine sample with non-specific gravity, and they will be you'll be given urinometer, and you'll be asked to measure that. Okay, then they will evaluate your performance, and also microscopic identifications. This is also very important. Okay, identification of, for example, cast, crystals, parasites. Okay, so we have to know what casts look like. Hyaline cast, maybe granular cast, waxy cast. Okay, we have to identify the type of cast that we usually see on under microscope, and also the crystals. Is it calcium oxalate monohydrate, dihydrate, burate? So we need to identify the type of crystals, and also parasites, such as the very common parasite is schistosoma hematobium, right? Schistosoma hematobium. So we have to identify the common casts and the common crystals. The common cast and the crystals. Okay, this is very common. This is very common. So another thing is we have to now the chemical strip tests and their interpretation. We may be asked to do a chemical test on urine sample. We may be given a strip, and we may be asked to measure the chemically the urine chemically, and we may be asked to interpret it. So the microscopic identification, the physical identification, and also the chemical examination is very important. We need to know how to do that, and also the interpretation and how to report it. The correct form of interpretation is also very important on the clinical movie exams. So this is all what I have about the clinical practicum session. So take this direction to prepare yourself for this exam. Thanks for watching until then. Don't forget to subscribe and share to your friends, and also like the video. So next we'll be back with another video. Until that, have a nice day.